hope you all grabbed breakfast, hope you all got coffee, and hope you are all ready for this morning. Good morning and welcome to the second day of the 39th Annual HDSA Convention. Thank you for that, and thanks for being here, and welcome to the HDSA Research Forum. It's good to see you all. Hope you're having a good time. I'm Dr. Eric Johnson. I'm the Chief Mission Officer here at HDSA. I want to start first and foremost by thanking the sponsors who make it possible for us to do this. So thanks to Teva, Genentech, Neurocrine Biosciences, Sage Therapeutics, Unicure, and Enroll HD and CHDI. Thank you guys for letting us do this. Thank you for making it possible. Thank you. And I'm going to clap over here because otherwise I'm going to do it right in the mic and you would hate me for that. Today we're going to spend some time focusing on HD science and research, but I want to take a few minutes to talk to you all about the HDSA mission programs and services. I'm sure you've heard this before, and I know you've heard it even just in the last day and a half that we've been here, that HDSA's tagline is help for today, hope for tomorrow. When we talk about that hope for tomorrow, that's the work that's being done by HD scientists and researchers. And for all of you that are participating in clinical trials, that hope piece is the work. It's not just wishing it's going to happen, it's the work that's being done to get us closer to having treatments for Huntington's disease, that ultimately get us closer to having a cure. But while that's being done, HDSA is working every single day to improve the lives of everyone living with HD and their families. And that's the help for today part. I'm sure you've heard this before, and I know that I say this to people, and then when I used to be in clinic, I would say it all the time, is at this time, there is not a cure for HD. That's not news, everybody knows that, but it still kind of sucks to hear, right? There might not be a cure for HD right now, but there is a lot that can be done to help people live better with Huntington's disease. Medications are available to treat the symptoms of HD. I mean, the fact that we have 3 medications approved by the FDA to treat the movement symptoms of Huntington's is really a big deal. We know there's more to be done, and that is happening. There are trials that are going on right now that are looking at addressing other symptoms of HD, and those medications are important. But there's other things that are available to folks too. Medications to treat mood symptoms. Even though someone says, well, I have Huntington's and I've got depression because I've got HD, guess what? The same pill that helps a person who has just depression helps the person who has depression and Huntington's disease. It breaks my heart when I hear people say, there's nothing they can do about HD because there isn't a cure. There are so many things that you can do, and there are so many people that want to help you do it. In fact, I kind of want to take a second and ask the following people to stand up for a minute. If you are a neurologist, a psychiatrist, a medical doctor, a DO, would you please stand up? Oh, hold on. There's going to be more. If you're a social worker, would you please stand up? If you're a psychologist or a neuropsychologist, would you please stand up? If you're a physical therapist, an occupational therapist, a speech or language pathologist, would you please stand up? If you're a dietitian or a nutritionist, stand up. If you're a nurse, please stand up. If you're on a research team, please stand up. If you're a clinical coordinator, please stand up. All of these people are part of the teams that are working every single day to make the lives of people living with Huntington's disease better. Thank you guys so much for everything you do, and thank you to the room for acknowledging them. When people say there's nothing I can do about HD, I would like to say BS, and I'm going to truncate it because I would say other things. All of those folks, everyone who just stood up, is here to help people living with HD. They're not going to be able to wave a magic wand. They're not going to be able to snap their fingers and make it go away. They might not be able to fix everything that a person is dealing with, but they can do a lot to help people live better with Huntington's disease. Going back to the HDSA mission programs and services, Whether led by the national level like my team at HDSA or out in the field by our HDSA social workers, the folks at the HDSA Centers of Excellence, the volunteers at our chapters and affiliates, there are a lot of different resources available to people living with Huntington's. I'm going to see, if y'all got a convention bag, you should have this card in there. I'm not going to go through it. This kind of hits on 10 of our high-level programs and services that people can access. This is a great resource if you're out in the community and someone's like, I'm living with HD, what can I do? Here's 10 things right here. Now, you might not have this card on you at all times, and that's where the beauty of that little QR code at the bottom is. It's like, hey, I scanned this on my phone. Here's the website I can show you. Or you can just tell people to go to the HDSA website, and I hate saying www dot anything just because it's hard, but that's the website, www.hdsa.org. You can find all this stuff. In our mission programs and services, they do fall into 7 areas: advocacy, clinical care, community services, disability, education, research, and youth and young adults. I hope you've noticed this, but the different sessions you've been going to over the course of this convention are all correlated with one of those 7 programs. So if you're sitting in a session and you're learning about how to advocate at a national level, that's going to correlate to our advocacy programs. So we wanted to do that just to kind of have you have the sense of, while it's great to learn in the room, there's more that you can follow up with later when you leave here today. For all those folks, I just remembered there's like people who are watching this, and for all of you who are out there watching this right now and just heard me say that, it's on the website. You can follow it all too. Man, no, my mom's watching, which is even worse, so I'm going to be getting a text that's like, you just said that. So forget where I was. The convention program lines up with our mission programs and services, so please follow along. You can see how those things relate to the talks that you're going to be listening to yesterday and today. Speaking of the convention program, I really want to thank the program planning committee. Convention doesn't just happen, right? We had a group of volunteers from Washington State, Oregon, Idaho come together, and over the course of 3 months in different meetings and different work that was done outside of those meetings, put together the recommendations for the sessions that showed up in this program. So I really want to give a thank you to anyone who volunteered on the program planning committee. Thank you for making this. I also want to thank all the experts who are presenting in our sessions today. And I'm using the term experts on purpose because everyone in this room, whether you are, at your first HDSA convention and you just learned about HDSA 2 weeks ago, or you're someone who's been working in this field since before Dr. Bird, you are an HD expert. You're an HD expert. I want to thank all the experts who have volunteered their time to present a session, to lead a support group, to work with as a volunteer in any of our exhibit hall booths or sessions. Thank you all for that. Please thank them for that. And finally, I want to thank the entire HDSA staff team. Putting together and pulling off a meeting like this is no small effort. And I hope that you've gotten to meet some of the staff around during the days yesterday and today, and if you haven't already done so, please do so today. Everyone's lovely, everyone is smiling, everyone is probably spinning around on the inside, but that's what happens when you bring together as many people as we have for this convention. And I just want to thank the entire staff. You guys kick butt, and I truly appreciate it. So please help me thank the staff. All right, thank you guys. Let's talk research. Huntington's disease research is vital for all of us to better understand the disease, to identify and ultimately develop treatments for the disease, because this is going to change the way that people live with Huntington's disease. In order for that to happen, we need people living with HD to participate. Without you, nothing happens. You heard this message from Louise previously. You're going to hear it again throughout the day. We need people living with HD to participate in the clinical trials that are out there, because if you don't participate in those clinical trials, the clinical trials go away. If clinical trials go away, potential therapeutics never move forward. We need people to participate. It's scary. There's things that have happened in just recent history that might make people say, I don't want to do that because, and we get that. But it takes a lot for a drug or a study to get to where it is. And while things do happen, it always happens. Can I say this? Shit happens in science. It does, and it's not always good stuff, but sometimes good things do. We've got those 3 drugs that are approved, right? But none of that would happen if people didn't participate. So I encourage you all to participate to your level of comfort, maybe push yourself a little outside that level of comfort if you can, because if you don't, nothing's gonna happen. There's some easier ways to do that, right? You can't necessarily just get in a clinical trial today, but you can participate in research today. If you've not done so already, our friends at CHDI, and I point that way because it's down that hall, and then you hang a right, there is an active research study happening here during convention, so please participate. You're gonna hear more about this. You've heard about it a lot. You've already seen it on the slides. You can participate in research from your phone, from your computer, from your laptop or your tablet, whatever the hell that thing is called, at home, right? The Power HD Natural History Study is a way that anyone living with HD can participate in research. You're going to hear more about it this afternoon. You can scan this QR code right now if you want to get the process started. You can talk to Tina and Carmen, who I think are in the room, and Caitlin, who might be outside the room. Yep, over there. You can go to the RareX booth that's right outside, and you should really pay attention when Jenny's up here later talking at lunch today about the Power HD study. It's easy. Ultimately, you might say, oh, dude, I did that initial consent form part. Get through it. The rest of it is easy. Talk to the folks out here. Just sign up today. You can scan this, create an account, and come back later. It takes 2 minutes to set up an account, make a password. Go and do the surveys later when you've got time. Be a part of it. You're the ones who are going to make things happen, and there's no better time to do it than now. Now I'm going to shut up, and, well, not quite yet. I'm going to finish that part. I'm very excited to introduce our speaker to you today, who's going to be talking to us about somatic expansion. This is an important concept in HD science, and one that you've likely been hearing about a lot recently. And I'm pleased to introduce Bob Hansaker, who is a staff scientist in Steve McCarroll's lab at Harvard Medical School and the Broad Institute. He holds a joint appointment at the Harvard Medical School Department of Genetics and the Broad Institute of MIT and Harvard. And you should be impressed by that. It's very cool. In that role, he's in the Medical and Population Genetics program and a member of the Stanley Center for Psychiatric Research. Bob's research focuses on the analysis of how the human genome varies at highly mutable sites, including large structural changes where entire copies of genes are gained or lost, as well as smaller DNA variation, including short tandem repeats, such as the CAG repeat that causes Huntington's disease. I know that was a lot of words that you're probably just like, I don't know what he just said, and he doesn't necessarily know everything he said either, but you're gonna learn more about it. Another theme in his research has been the analysis of somatic mutation, including both large and small genetic changes. Finally, Bob is the author of the Genome Strip software, which is a genome structure in populations software platform. It's a suite of tools for discovering and genotyping structural variations using sequencing data for computational analysis of structural variation. I can barely make a PowerPoint, y'all. He built a whole system. This is all really awesome, but Bob is also a real human being too. In fact, he attended high school here in Battleground, Washington, near the foot of Mount St. Helens, before heading east to study at MIT. So please join me in welcoming Bob Hansaker to the stage.
All right. Well, thank you so much. I'm really excited and honored to be here and to be able to share with you today some of the recent work that we've been doing on Huntington's disease. I work in Steve McCarroll's lab at Harvard Medical School, and this work has all been jointly done with the lab of Sabina Beretta, who's at Harvard Medical School and also at McLean Hospital. So I've got to make sure I can run the clicker here. All right, so I do have one small financial disclosure. My employer, the Broad Institute, has filed patent applications. Oops, sorry about that. Has filed some patent applications related to portions of this work. So for this audience, I'm not going to start with a description of the symptoms or prevalence of Huntington's disease. Maybe the only thing I will say is that although historically Huntington's was described primarily as a movement disorder, as Eric just mentioned, we know today that there's a whole variety of different symptoms related to Huntington's disease, some more subtle and some that affect different people in different ways at different stages of the disease. So it's perhaps more complex than George Huntington realized back in the day. So one of the themes I'm going to talk about today is disease progression. And what I mean by disease progression is, for somebody who has HD, there was one event that happened. At birth, they inherited a long HD-causing allele. And then later, decades later, if it's adult onset HD, symptoms manifest. And so in between those two things, there must be some causal chain of events that happen to get from point A to point B. And that's not terribly well understood today, what all those steps are to get from A to B. But what I'm going to suggest is that the more we can understand about that causal chain of events, the better position we'll be in to fight against HD. So we know that one of the links in the chain is that HD pathology involves the death of specific kinds of neurons in specific brain areas. And while only certain types of neurons are lost, these neurons are abundant and they're very important. You can see on this MRI scan here the atrophy that results from the loss of these neurons. So a principal, whoops, I need to fix one thing here. Or I need to be more careful. So a principal type of neuron that's lost in HD is what are called striatal projection neurons, or SPNs. And sometimes in the literature, you'll also see these referred to as medium spiny neurons, or SPNs. I'm going to talk a lot about SPNs today, and precisely because they're one of the types of neurons that's most dramatically affected in HD, particularly when it's in an early symptomatic phase. So we know a lot also about an early link in the chain, which is the inherited HD-causing allele. And we learned much of this from studying large families with HD, including families from around Lake Maracaibo in Venezuela. Today, HD is quite literally the textbook Mendelian genetic disease. It exhibits autosomal dominant inheritance and full penetrance. In 1993, as you probably know, it became the first autosomal dominant human disease for which the responsible gene was found by genetic mapping. And at the time, this generated tremendous excitement. It was one of the things that encouraged us to sequence the whole human genome. We know that all Huntington's patients have an expanded trinucleotide repeat in which the DNA sequence CAG is repeated a variable number of times. And this CAG repeat is in the first exon of a gene called Huntington, which is abbreviated. And I'm sure you know that most people in the world have CAG repeats with less than 30 copies of this CAG sequence. But if you inherit even one allele with more than 35 CAGs, this of course leads to Huntington's disease. Moreover, the length of the inherited allele impacts the age of symptom onset. The longer the allele you inherit, the earlier the age at which symptoms tend to first appear. So we know in some sense what this expanded CAG repeat does. What it does is it creates a longer HTT protein that contains some extra glutamines. And the prevailing assumption has been that there's something about this longer protein that is mildly toxic, perhaps right from birth, and that this toxicity builds up over decades and decades to lead to symptomatic disease. But new data I'm going to tell you about has caused us to rethink this model a little bit. The HD-causing allele might still create a toxic protein, but maybe not quite in the same way that we thought. The new data suggests that maybe there's a key link in this causal chain of events that involves somatic expansion of the HTT-CAG repeat. So what is somatic expansion? When we're born, we inherit DNA in the form of one set of chromosomes from our mother and one set of chromosomes from our father. And together, these make up our inherited genome. But in every cell in your body, inside the nucleus, that cell has its own private copy of your inherited genome. You have a lot of cells in your body. That's a lot of copies of your inherited DNA. And it turns out, not all of those copies are 100% identical. Whoops. And so when those copies of the DNAs in different cells are not exactly identical, we call this somatic mosaicism. So some cells have slightly different DNA than other cells. And often, somatic mosaicism can be like an invisible phenomenon, but sometimes it can be made visible. For example, if it affects a phenotype like pigmentation, right? It can cause something that's visible on the outside, like this dog, right, has a somatic mutation that's created a different patch of their fur that has a different color. And this can happen in humans too. We can have somatic mosaicism that affects pigmentation. In the context of HD, when we talk about a somatic expansion, what we're talking about is somatic mosaicism, but specifically about the HTT-CAG repeat. And specifically, if that HTT-CAG repeat is now different than the inherited length and is differing from cell to cell, especially in neurons in the brain. So we've known for a long time that the HTT-CAG repeat can mutate somatically in HD patients. Peggy Shelburn's lab showed 20 years ago that if you isolate DNA from the brain tissue of HD patients, that DNA contains molecules with a wide range of expanded CAG repeats. In this work, they also found that the amount of somatic expansion varied across brain regions. And a number of other researchers have made similar observations, both in humans and in mouse models of HD. So longer inherited alleles result in earlier symptom onset, on average. But there's actually a lot of variation from individual to individual. If you focus just on people who have an inherited allele that's, say, 42 CAGs, well, some of them will have an age of onset that's right around the average for someone with 42 CAGs. But some might be 15, even 20 years earlier. Some might have onset 15, even 20 years later. And in between those two extremes, there's a whole bell curve of people with different ages of onset. So the GEM-HD consortium has done a series of increasingly large genome-wide association studies to see whether there are other places in the genome, other genes, that act as modifiers for age of onset. And the results of these studies have been really striking. Many of the modifier genes turn out to encode DNA maintenance proteins. And several of these proteins have now been shown to regulate the stability of DNA repeats, like the CAG repeat in HTT. Functional studies by Vanessa Wheeler and Ricardo Moro-Pinto and others have shown that these DNA maintenance proteins also affect the rate of somatic expansion directly through experiments in mice, suggesting that somatic expansion may be the mechanism by which these modifier variants are affecting age at onset. And Vanessa Wheeler's lab has also observed that HD patients with earlier disease onset not only carry longer inherited alleles, but they also seem to exhibit more somatic expansion. But there are fundamental biological questions about HD that remain unanswered. What makes the inherited HTT alleles neurotoxic? Why does neurodegeneration take decades and decades? And why is it so specific to certain types of neurons, and yet it doesn't affect others? So we wanted to try to answer these questions, and so we teamed up with Sabina Beretta and her lab at McLean Hospital, and she also runs the Harvard Brain Tissue Resource Center. And we wanted to analyze postmortem brain tissue from HD patients and controls that don't have HD using single-cell RNA sequencing. So single-cell RNA sequencing is a relatively new technology. One of the first versions of this technology, called DropSeq, was actually developed in our lab between 2011 and 2015, led by a postdoc named Evan McCosco. And the power of single-cell RNA sequencing is that it lets you measure which genes are turned on or turned off in individual cells, not in the whole tissue, but in each specific cell. But we faced a challenge, which is that the genetics suggests that somatic expansion is likely to be involved somehow in HD. And while the RNA sequencing could tell us about which genes are turned on and off, we lacked any accurate way to measure the potentially expanded CAG repeat in the individual cells. So over several years, around 2020 to 2023, we developed a new laboratory approach, excuse me, to allow us to measure both the gene expression from the RNA sequencing as well as the repeat length in these individual cells. And we do this by running both a standard genome-wide RNA sequencing protocol plus a second custom protocol we designed that specifically targets the long CAG repeat in Huntington exon 1. And as part of this, we used a new technology called long-read sequencing to do the repeat length measurements. And this allows us to accurately measure the CAG repeat length from an individual cell, even if it's been greatly expanded. And together, these two analyses are linked together by these cellular barcodes. So we get to match up the data, and it leads us to be able to do some really powerful downstream analyses. So armed with this new technology, we began to look at HD at the single cell level, focusing initially on the caudate. And we first looked at which cells are lost and which are not lost in different people in different stages of disease. So on the left, the colored bars are showing the proportion of the different types of cells in different people where we sampled their brain tissue from the caudate. On the top are the controls. And you can see the SPNs, which are the pink bars over at the right. They have about the same length in all the controls. There's some variation. And then when we get to the bottom, we have the people with HD. And they're ordered here by their CAP score. And CAP score is based on the product of their age and their inherited allele length. And it's a pretty good proxy for the stage of disease progression. And you can see that in the people with HD, the pink bars are shorter and shorter. Right? There's fewer and fewer SPNs as we look at people with higher CAP scores. And you can also see that on this plot where it's more directly shown against CAP score, that the higher somebody's CAP score, when we measure the cell composition in their caudate, we see that they have fewer and fewer SPNs. From this new data, we're also able to see that the somatic expansion is exquisitely cell type specific. Here I'm showing the data from brain tissue from one donor from their caudate, and we see profound somatic expansion only in the SPNs, not in the other cell types present in the caudate, where there's very little somatic expansion. And we found this to be true not just in one donor, but across every donor we looked at. The cell types where we're seeing lots of somatic expansion are precisely the same cell types that are the vulnerable neurons in HD. And our new assay allows us to see extremely long CAG repeat expansions, previously not detectable at this scale. We observed some neurons with up to 1,000 CAG repeats. This is from somebody who was born with probably 42 or 43. And this is confirming those early observations from Peggy Shelburn's lab. And we also noticed something else that intrigued us, which was that the length distributions of these repeat expansions had a really unusual shape. While the majority of the cells have undergone only modest expansion, up to like, say, 80 or 100 CAGs, there was always, in people who had symptomatic HD, a small fraction of cells that make this really long tail, where they've expanded up into hundreds of CAGs. And this was really surprising to us. And also, like, I talk about this now as like modest expansion up to 80 CAGs. I think when we started this work, we would have thought 5 CAGs was pretty good expansion, or 10. But now we think of like a single neuron expanding to 80 CAGs as kind of modest, right? Long expansion we think of today as hundreds of CAGs in the repeat that's in that individual neuron. So, intrigued by the shape of these distributions, and we wanted to understand the temporal dynamics that might drive this pattern of somatic expansion, because we saw this armadillo shape in multiple donors. And if you plot that armadillo shape a little bit different way, this is called a cumulative distribution plot, you can see that there's kind of this weird kink. And this is not something you often see in biology. The kink is right here. And it happened in every donor. And it happened at right about the same CAG repeat length, somewhere around 70 to 90 CAGs, probably centered at about 80. And we were really intrigued by what could create like this shape in the data. And so we worked on using some mathematical models and some computer simulations to try to figure out what sort of dynamic process could create this kind of data that that we're observing from these people's brains. So we sought to base these models on some realistic biological assumptions about what's known about the biological mechanism behind somatic expansion. And these models use as input the observed repeat lengths from the data, the age of the donor when they were sampled, the donor's inherited allele length, and an estimate of how many SPNs they've lost based on the sampling of their caudate. And our modeling suggests that there are actually two relatively distinct phases of somatic expansion. The data suggests that the first phase is slow. It takes many decades for the average neuron to expand from 40 up to about 80 CAGs. But then the second phase is fast. Once that neuron reaches about 80 or 90 CAGs, the rate of expansion dramatically speeds up so that the neuron ends up with hundreds of CAGs in the repeat within only a few years. So on the right, I'm showing an animated simulation for one particular donor that shows the predicted distribution of repeat lengths over that donor's lifetime, along with the predicted fraction of SPNs that have been lost at that point in time. And these models ended up fitting the data really well, and they generate the shapes we're observing in the data. And an important feature of these models is that they predict that the individual neurons enter this rapid expansion phase asynchronously across a long period of time. So they don't all start rapidly expanding at once. It happens at different times. And almost none of the neurons will even begin this process until the patients are several decades old. We also did measurements on tissue from two donors who inherited a disease-causing HD allele. It was along a CAG repeat, but they were both pre-symptomatic when they passed away. And these two donors are shown on they're the two donors on the top of this plot. And for comparison, I'm showing you a donor on the bottom who had symptomatic HD when they died. And what we see here is very consistent with this model of the slow somatic expansion followed by the fast somatic expansion. The donors that were pre-symptomatic, they've had a lot of SPNs that expanded to like 70 or 80 CAGs, maybe up to 100, but only a very few cells that got over 100 to 200 CAGs. So this is giving us a model for the temporal trajectory of somatic expansion in individual SPNs. There appears to be this decades-long phase of slow expansion, followed by more rapid expansion. And I was actually telling Eric before I got up here that last night I went to the talent show, and something that surprised me a little bit was how many people I observed that had juvenile HD, which we don't always see in the general population. And so you'll have to forgive me if I have this tendency, if I talk about like HD generically. In my mind, I'm mostly thinking about adult HD. And not so much about juvenile HD, because in this study, actually, we didn't have the ability to study anyone with juvenile HD, although that would be a great thing to do in the future. So because we're doing okay on time, I'm going to also do like a small digression here, which I debated about doing. Because it's a little bit in detail, but bear with me here. But I'm going to talk a little bit, because I think maybe it'll be interesting. What is the mechanism that underlies it? So I've talked about how somatic expansion is this idea that the neurons have different repeat lengths. Each neuron has, in its own private copy of the DNA, a slightly different repeat length. But how did it get there, right? So there's a great deal of work that has been done on this, trying to understand the detailed biochemical mechanism, not by our lab, but other people like Anna Plechenik and Kara Usden. But I'm going to give you my explanation of my understanding of the current consensus in the field of how we think this process works. So as you know, the structure of DNA is a double helix, which is kind of like, you can think of it like a twisted rope, right? But it's not just a twisted rope. It's also like a zipper, because the DNA letters, the CAGs and so on, are on each strand of the rope, and they actually lock together. The C's pair with the G's, the A's pair with the T's, and these fit like a key in a lock, and sort of like a zipper. So DNA is actually kind of like a twisted zipper in its structure. And most of the time, and so occasionally, cells actually need to untwist and unzip the DNA. And they do this like locally and temporarily. For example, to transcribe the HTT gene to make an RNA, the cell needs to untwist and unzip the DNA briefly. And when this happens, the cell unzips the DNA, then the DNA zips back up, and almost always the DNA zips back together perfectly. But we think what underlies the mechanism that creates somatic expansion is once in a great while, that zipper doesn't go back together the right way. So I actually borrowed this image from the online Jeep Wrangler forum because it turns out that the soft tops on certain Jeeps actually have a similar problem, that their zippers don't always go together, right? And the idea is when you get a long CAG repeat, now the zipper can slip a little bit. And it'll create this little, what's called the slip-out structure. It's kind of like a little bubble in the DNA. Sorry, I keep fumbling with this a little bit. So here's a diagram that's showing a CAG repeat with two of these slip-out structures that are complementary because the zipper didn't quite go back together, right? And there's actually nothing wrong with the DNA at this point. It's just zipped a little bit incorrectly. And if the cell just unzipped the DNA and rezipped it, almost certainly it would zip up fine the next time. But while the DNA is in this transient state, the problem is that these DNA maintenance enzymes, they recognize the slip-out structure. This slip-out structure in this case is created by miszipping, but a mutation could also have created a slip-out structure like this. And the job of those DNA maintenance proteins is to monitor the genome and find mutations and fix them. So that if the DNA maintenance proteins see that slip-out structure, they say, aha, we have something to do, we need to fix this DNA. And they actually fix the DNA by snipping one side of the zipper, they peel the DNA back, they kind of chew it back, and then they resynthesize it using the other half of the zipper as a template, the other strand of the DNA. And when they do this, in this case of mistaken identity, because they thought there was a problem that needed to be fixed, this can add an extra CAG to the repeat that's in that cell. And we think this is the underlying mechanism that causes somatic expansion of the repeat and why it happens only at repetitive sequences like CAGs and only when they get long, like more than 35 or 36 bases. I'm sorry, 35 or 36 repeat units. So I've talked a lot about somatic expansion, the dynamics of it, the new things we're able to measure. But there's this fundamental question of whether or not somatic expansion actually drives HD pathology. Is it part of this causal chain of events? So this is actually not an easy question. If you were just to do a simple comparison of gene expression between people who have HD and people who don't, I'll refer to them probably as cases and controls, we see thousands of differentially expressed genes. And that's represented here in these volcano plots. Volcano plots, there's a dot for each gene, and it's telling you whether that gene looks different between one group and another, here between people with HD and people not with HD. The colorful dots are ones where there's a lot of statistical power to tell that the gene is really different, and the color tells you whether that gene is higher in one group versus the other, or whether it went down in one group versus the other. And the point here is that there's thousands and thousands of changes. If you just compare a person with HD to a person without HD, looking at the gene expression and their caudate, and the gene expression changes are across all the cell types, not just the ones that are lost in HD. And we think this just reflects the fact that that if a person has symptomatic HD, their brain is in a very different state with someone who's never experienced HD. There's been cell loss, there's been atrophy, there's been inflammation. The whole brain, which is a tightly interconnected organ, is reacting to this. So all the genes, all the different cells are expressing genes in a slightly different way, in a slightly different pattern. And this creates a problem, which is that we have to be able to separate the causes from the effects, right? Because it's like looking for a needle in a haystack because there's so many different changes in an HD brain. But we realized that there was a way we could approach answering this question. So as we've seen, in any one tissue sample from a single donor, there are thousands of SPNs present. And these SPNs, it turns out, they all have different repeat lengths. Some are at 50 CAGs, some are at 80, some are way up in the hundreds. So rather than comparing someone with HD to someone without HD, we can compare the neurons within the tissue from a single donor and ask what happened to those neurons as that repeat expanded. And this gives us a powerful way to control for external factors and zero in on exactly what the effect of the somatic expansion is. So if we do this, and we do this kind of by we did this initially by sort of looking at SPNs that have just like mild expansion, kind of medium expansion, and maybe really long expansion. And if you just take the like slightly expanded SPNs and the medium expanded SPNs, like between 66 and 150, and you ask, like, how did the gene expression change? We got a really surprising result, which is that there weren't any changes, right? As that repeat expanded from 40 to 50 to 80 CAGs, we could detect no changes in the gene expression in the neurons. But then we thought, well, what if we look at that small percentage of neurons that have repeats that have gone way up into the hundreds? What would we see if we looked at the gene expression of those and compared them to all the ones that were shorter? And when we did this, we began to see hundreds of genes where the gene expression was changing as the repeat got longer. But it only began to kick in when the repeat got to about 150 CAGs. And we actually did this analysis a couple of different ways. I'm going to go through one here that's a little bit more detailed. What's this very colorful blob at the top is what we call a heat map. And there's actually many little colored dots on there. And each row in the heat map is a gene. There's about 400 genes represented here. Each column in the heat map is a single cell, a single SPN. And the color that's in each dot is telling you whether or not that cell is expressing more or less of the particular gene. And you can see that the color is kind of a mishmash across most of the left side of the plot, but then there begins to be a pattern in the color over to the right. And the next two rows are sort of summarizing this data in terms of in each cell, if we take all these genes and ask on average what's happening to these particular sets of genes, and we pick these genes because in the longest in the SPNs with the longest repeat lengths, they were the ones that were dysregulated. And we are dividing them here into the ones that like to go up when the repeat gets long, those are in blue, and the ones that like to go down when the repeat gets long, and those are in orange. What you can see here is that the expression of those genes is really, really stable across this whole spectrum of repeat lengths until you get to about 150. And then interestingly, once you get to 150, the expression begins to change, and the longer the repeat gets, the more the expression continues to change in the same direction. The gene expression is getting more and more distorted in the same direction. And this pattern of gene expression changes was very consistent across donors. Here's data we sampled from 6 different donors, different disease stages, and in the CAUD data of every single donor, when the SPNs got to about 150 CAGs, this set of genes began to have distorted gene expression. And we have begun calling these the continuous changing genes, because the gene expression changes start at about 150 CAGs, and then they intensify with increasing repeat length, but only after we've crossed that threshold. So the progressive dysregulation of these genes represents a third phase of SPN pathology at the single cell level, and we've begun to call this phase C. And we observed that these dysregulated genes have some specific properties. First of all, the phase C genes are highly expressed in SPNs, and the genes that go down in phase C are strong markers of SPN identity. They make SPNs look different than other types of neurons, whereas the genes that go up are broadly expressed across neuronal genes. So the net effect is that the phase C genes are kind of an identity softening of the SPNs. They don't look quite as much like SPNs as a healthy SPN that hasn't had the repeat expand this far. And this has also been described a little bit as the SPNs may be looking like they're in a little bit less mature form. They're regressing a little bit. So this is the third phase of our trajectory of HD pathology in an individual SPN. First, the cell undergoes this slow repeat expansion, then it goes through a phase of faster repeat expansion, but with no cell-autonomous gene expression changes happening. Until we get to the third phase, which we call phase C, hundreds of genes become dysregulated, and this dysregulation increases as the repeat expands. We also saw a second group of genes that had dysregulated gene expression when the repeats got long, but they had a different pattern than these continuously changing genes in phase C. So we began to call these the phase D genes. One of the hallmarks of the phase D genes is that they're normally completely turned off in mature SPNs. You never see these genes on. And when we see these genes start to come on, they come on as a group. And they come on in a specific SPN as a group, and they tend to happen in SPNs that have these very long repeat lengths, but there doesn't seem to be a specific threshold. And that's illustrated here in these two charts, which are showing you how likely it is that the phase D genes would turn on in an SPN based on either how long the repeat has gotten or how much of the phase C changes that SPN has undergone before we get to phase D. And interestingly, some of the genes that are turned on in phase D are genes that have been associated in other studies with things like the cell beginning to shut down or to go to sleep or to stop doing its job. And so what we think these phase D genes represent is this kind of terminal phase of the cell starting to shut down. We sometimes call phase D the de-repression crisis, because all these genes that normally shouldn't be turned on are turned on. And another way I sort of think about this is that we think that maybe phase C represents that now the HTT protein has finally become toxic for some reason. When it's gotten to 150 CAGs, the cell begins to feel the effect of this toxicity. It's struggling against this toxic effect. The phase C genes are beginning to become dysregulated, and the phase D genes represent, in some sense, the cell losing the struggle against this toxic insult that it's begun to feel. So when we began this work, we expected to see toxic effects from a mutant Huntington protein that would start very early and then gradually increase over a patient's lifetime. But instead, these experiments had led us to a very different model of Huntington's disease pathology. We now think that the pathology begins with a slow phase that lasts decades, kind of like a slowly ticking DNA clock that's proceeding asynchronously in individual neurons. But once a neuron reaches about 80 CAGs, the clock speeds up. And then when the repeat reaches about 150 CAGs, the cell enters this period of gene dysregulation, leading to cell death, probably within a matter of just months. Just realized my time has been flying by here. So we know that there's a dramatic loss of SPNs in the later stages of Huntington's disease, and we can represent this as a fifth phase of SPN pathology at the single cell level. So we call this phase E, and this is the phase in which the SPN has been lost. And if we combine these 5 phases with the computational simulations that I showed you earlier, this gives us a framework for predicting from a donor's data how many SPNs were in each of these 5 phases over the course of a donor's lifetime. And here's the model for one such donor with a typical inherited allele of 42 CAGs. In the early years of this donor's life, when they were up to, say, their early 20s, the model predicts that almost all of the donor's SPNs would be in phase A, the slow expansion phase. And as the donor approaches middle age, they begin to experience slow loss of SPNs. The trajectory of this loss of SPNs then goes on to become faster over time. And an important implication of this model, especially in the context of therapeutic intervention, is that at any given point in time, the majority of the existing SPNs are still in phase A, the slow expansion phase. Only a small percentage of the cells at any given time are in phases C and D, the phases in which we think their mutant Huntington is causing problems. So we can also investigate what the models would predict relative to milestones like symptom onset. And one caution, however, here is that so far we've only deeply analyzed a few donors, and for some of these the medical records are sparser than we would like. But with that caveat, we do see that motor symptom onset tends to fall in the range of about 10% SPN loss, up to as much as maybe 40 or 50%, although we worry that that number could be biased upwards by having incomplete medical histories. But this does begin to give us a picture of how events at the cellular level may correspond to external milestones in the progression of disease. Thank you for that. I wanted a little mood music. So our model of asynchronous pathology suggests we can have a little different mood. I wanted a different mood for this slide, actually. So our model suggests some surprising answers. We want some surprising music to long-standing questions about HD pathology. So what is neurotoxic about a typical inherited allele with between 36 and, say, 100 CAGs? Well, perhaps nothing, Perhaps these alleles are in themselves actually not neurotoxic at all. And why does neurodegeneration take decades? We actually think it doesn't. What takes decades is for an individual neuron to reach 150 or more CAGs. But once neurodegeneration starts, it's a rapid process for that neuron. And why is the degeneration so neuron type specific? We believe this is because somatic expansion rates vary across the different cell types. And so the repeat only reaches this high threshold of 150 CAGs in a few vulnerable cell types. Overall, we estimate that HD pathogenesis is a DNA process based on somatic expansion for more than 95% of a neuron's life. So what are the implications for HD therapeutics? Well, first of all, I think knowledge is power. We believe that a deeper understanding of the disease process will have a cascading effect to accelerate progress against HD. More complete understanding can potentially unlock new therapeutic opportunities, including therapies with different modes of action, such as targeting somatic expansion directly. And these therapies could potentially then be used in different contexts or in combinations. HD neuropathogenesis appears to be a transcriptionally silent, DNA-based process for more than 95% of a neuron's life. And this suggests that there's a potentially large therapeutic window for intervention, including before symptoms commence. And then lastly, at any given point in time, most of an HD patient's neurons have repeat lengths that are below 150 CAGs. So this means that there's also a good therapeutic treatment window, and this doesn't close when symptoms begin. So this work is now available as a preprint. I will note that it has not been certified by peer review. You can find it by Googling, or if you want, you can take a picture. I put the QR code up here nice and big if you want to read the preprint. If you do read it, we would love to get your feedback. And yes, that's you, the families, and the members of the HD community. So we're pretty much at time, right? So I'm going to skip a little section I had on follow-up experiments we've done in the cortex. The punchline is that we're doing a bunch of these experiments now, not in the caudate, but in the cerebral cortex, and we're finding that the same basic pattern holds in the cortex as in the caudate. There are vulnerable cell types, there's somatic expansion that goes way up to 150 CAGs and more. The genes begin to become dysregulated. We see phase C changes and phase D changes in the cortex, just like in the caudate. So I'm going to fast forward through that to my acknowledgement slides here. So the conclusions I pretty much already said, that the pathology appears only after about 150 repeat units. Individual neurons experience this toxic mutant Huntington asynchronously over months and not decades, and that ANHD appears to be a DNA process for most of a neuron's life. So this is the work of a whole team of people in Steve McCarroll's lab. Oh, you know what? I forgot to advance these slides. In Steve McCarroll's lab and Sabine and Beretta's lab, I want to acknowledge all of my colleagues. I want to acknowledge our collaboration with and our generous support from the CHDI Foundation. And I especially want to thank the brain donors and their families, because absolutely none of this work would have been possible without their generosity. I understand there's a session later today on brain donation, which I'm very interested in going to. I haven't signed up for brain donation yet, but I am like so sold on what a gift brain donation is for the future. So thank you very much. I'm happy to take it.
Bob, thank you so much for that. We've got a few minutes for questions if people have them. There's some microphones running around the room. I can't see a thing, so if you've got a question, just holler into your mic and let us know.
Good morning. I'm Christy Figueroa Perez from San Diego,
wave
California. Woo!
Okay.
So my question is, and I just want to make sure I'm understanding correctly, is between C and D, there's a progressive change that's really, right, progressive. Should we be retested at that time to see at what point we're reaching 150, how fast we're progressing, or do we just stay as we are and just go with the flow? Because that's what we've normally done.
Yeah, that's a great question. Today, there is no way we can test on a living person to see where they are in this trajectory of somatic expansion. We just don't have a technology to do that. So part of the reason we do the modeling is to sort of be able to work backwards, and an active area of research is to try to tie what we think is going on at the cellular level to external milestones that we can measure, like symptom onset, or maybe symptoms that are being recorded on like cell phone apps and stuff, reaction time and cognitive tests and things. We'd love in the future to be able to relate that to the underlying cellular changes. But no, there's no way we can test today for exactly where you are with respect to somatic expansion. Although I've had a conversation recently with CHDI about whether or not that could be done, like with an MRI or something, as a biomarker to be used in clinical trials.
Actually, Deborah, please hold for a sec. Is it a simple thing to say you don't need to retest because the CAG repeat in your blood is going to stay the same? The numbers that Bob is showing us really only comes from when we are able to look at the cells in your brain, which we can't do until you're dead.
That's pretty right, Erica.
We don't want to be taking cells from your brain while you still need it, please.
Here's my is it on? Maybe you echoed. It's on. Here's my question. Okay, we can't biopsy a brain, but we can biopsy a liver. So what if the process of somatic expanding the liver could map to the process in the SPNs? And you could know, well, if this is happening in the liver now, 10 years from now, it's going to start happening in the SPNs or something like that. We could have a biomarker based on a biopsy.
Yes, and there's a lot of interest and excitement about doing this. We haven't done any liver samples yet, but people have asked if we can run liver samples. Liver biopsy is one thing. I mean, something I meant to emphasize is that an interesting thing about somatic expansion is it's exquisitely cell type specific. It happens in the brain, but certain cells in the brain. It happens in the liver. I think we know it happens, for sure it happens in mice in the liver cells for somatic expansion. There's a little bit of somatic expansion in the blood. And so Darren Monkton and other people are working to use somatic expansion in the blood as a biomarker. For example, if you had a therapeutic that was supposed to reduce somatic expansion, could you detect the effect of the drug in the blood as a surrogate? And then people are also talking about, could you detect it with a liver biopsy? So these are all really exciting developments to me because we're talking about like biomarkers for therapeutic intervention, which is great.
Do we have one? Oh, hey, we'll take your last question here. Bob is going to be around
all day.
All day. If you catch him, he'll answer anything.
I'll talk your ear off, as a matter of fact.
Yeah, watch out. Yes, ma'am.
My question is based on your, the models and everything with the expansion and how it starts differently in different people. And this is considering adult HD. Is it possible to see early symptoms in teenagers?
So an interesting thing that our model, so by teenagers, do you mean juvenile HD?
Well,
or
no.
Adult onset HD, but when the person is a teenager?
No, I mean, looking back, we've seen things in our adult family, but can we watch out for things in our teenagers that may eventually get adult Huntington's?
I don't know if this would suggest particular things to watch for in the teenagers that you don't already know about. One interesting thing about the modeling here is that there's been kind of this mystery. I can't study that works on, but they've tracked children who inherited a long HD allele right from birth. They've done tons of testing on them. Some of them are in their early 20s now. And essentially the punchline is they look neurotypical for early 20-year-olds. And our models would predict that's exactly right? That you wouldn't actually see any changes as a teenager because your neurons are actually undergoing somatic expansion, but they don't seem to mind as long as it doesn't get really long.
Thank you, Bob. Thank you all for your questions. Please catch Bob over the course of the day. We're thrilled to have you here. Thanks for spending the day with us. I'm going to ask you to jump off the stage over here because your mic is going to get pirated real quick. Thank you all. And now I'm not going to say a heck of a lot more. I'm going to turn this over to the HD Buzz team. So please welcome them to the stage, or not quite yet, or here's a clicker. I'm going to get out of the way. They're going to do something. Welcome HD Buzz.